Hello there, this is Mrs. Scoville, and today we're going to be learning about meiosis. We are still in cell replication. Okay, there are three objectives for today's lesson. The first is to define what this thing meiosis is and explain why it is essential. We're also going to need to define the terms haploid and diploid to be able to understand meiosis and identify the stages of meiosis by description and by looking at pictures. Okay, so first we're going to start with kind of a broad reason that our bodies need to do meiosis. So when you have an organism like a human, it's going to have a predictable number of chromosomes. So here I've told you all humans have a total of 46 chromosomes. And in order for a species to continue and to mate, all members in that species need to have that same number of chromosomes. That's pretty much how species can mate. If they have the same number, they can mate. If they don't, well, they can't, which is why it's kind of funny, but like a goldfish can't mate with a trout. They don't have the same number of chromosomes. So because of that, we need to have specialized cells, and in humans it's egg and sperm, that actually have half the number of chromosomes. What this accomplishes is when they come together, egg and sperm coming together, that gives the 46 that we need for each individual to have. If we didn't have this mechanism to kind of get half the number, every single generation would have twice the number and they could never mate with the previous generation. This will cause the species to die out, and this is not what we want. So instead, we're going to use this process called meiosis. So a little bit more into this, exactly definition-wise, what meiosis is. Besides it just being a way to get half the number of chromosomes, technically it's a nuclear division. So with mitosis, we had a division of DNA where we were trying to make it so that cells were identical. Here, the goal is to reduce the number of chromosomes so that we have half. And we have a couple of terms to use for those to describe that. So a normal body cell we describe as diploid, meaning it has two copies of a chromosome. Our goal in meiosis is to get these haploid cells that only have one copy. And I've sort of already mentioned this, but in humans, we call them gametes, which are egg and sperm, or reproductive cells. We'll go through meiosis. This allows sexual reproduction to occur and keeps our number of chromosomes the same in all people. Okay, so this cell has this has a lot of text on it. I know that, but this really needs to drive home the point of diploid versus haploid. So starting over on the diploid side. Again, this is just a cell that has two sets of chromosomes, and we call those chromosomes homologous because they're going to have similar genes and similar size. So one of these homologous chromosomes will come from mom, and the other will come from dad. And this is for all people. And again, because you have one from mom and one from dad, you basically have two copies of each gene. With diploid cells, we'll represent these with the symbol 2N. So we use the symbol 2N, and in humans, this is going to be what are called somatic, or our body cells, and this is basically all of your cells in your body besides egg and sperm. So in humans, the diploid number would be 46, and you can also write this as 2N because that means diploid equals 46. So haploid is going to be the opposite of this, but again, just to recap, diploid, di, meaning two, you have two sets of these homologous chromosomes, one from mom, one from dad, overall two copies of everything. Haploid though, haploid, it's like half. You have a single set of all your chromosomes. So in a haploid cell. So in egg and sperm, each of the chromosomes are going to be basically either from mother or from father, and it'll have one copy of every gene. So here, we represent a diploid with 2N. We're going to represent haploid with 1N. So if you see something like 1N equals, that means that's the haploid number. In humans, egg and sperm are the only haploid cells, and therefore our haploid number would be 23 because it's half. And again, so we would say 1N equals 23. So if we look at this pictorially, this is actually called a karyotype here, and they can actually stain all of the chromosomes. So you can see in a diploid cell, there's a double set of all of them, whereas in a haploid cell, there's only half. And just a note here, the X, the fact that there's two of them, this is actually a female, we can tell. But again, diploid versus haploid, it just means the number of sets of chromosomes that you have. All right, so we need to know these terms diploid and haploid because in meiosis, one diploid or 2N cell is going to divide through a couple of rounds of division, and it will yield 4 haploid or 1N cells. And every single one of these haploid cells is going to be different than that original cell. This is not a bad thing. In mitosis, that was a bad thing. It led to cancer. But here, this is a good thing. It leads to genetic variation in offspring, which is why all people don't look the same. And as we already mentioned, this is very important to keep the number of chromosomes the same from generation to generation and allow a species to mate. Before meiosis, though, just like with mitosis, we are going to have DNA replication occur before. So here I've said it's characterized by one DNA replication followed by 2 successive nuclear and cellular divisions. So this is going to be a big difference from what we saw before. We're going to have 2 rounds of division. It's divided into meiosis 1 and meiosis 2. So the one thing I do want to point out to you here is this DNA replication is going to look slightly different because it will occur and it will give us duplicated chromosomes with sister chromatids just like we had with mitosis before, but we have a double set. So you're going to end up basically with, whereas before we had like 1 set of duplicated chromosomes, we'll have 2 sets of duplicated chromosomes. And again, we call these homologous or homolog chromosomes. So what that means is, for example, this maybe is the one from dad, this is the one from mom. They get duplicated before meiosis gets going. So, and that would happen during interphase 1. So this still is going to happen during interphase. Okay, so a little bit more into the stages of meiosis because that was one of our main objectives here. We're just going to talk about in words the kind of hallmarks, and then we'll go through individual stages. So in meiosis or meiosis 1, there's going to be a couple of key hallmarks that happen. The first is there's going to be this very important event called crossing over, and this is going to lead to genetic variation, which actually we'll get more into that in another lesson. And already right away we're going to end up with 2 daughter cells that are different than the original. We will still have that PMAT sequence, but here each one is going to have a 1 after it to denote that this is happening in meiosis. So prophase 1, metaphase 1, anaphase 1, and telophase 1, still in that order. Meiosis 2 is going to be very, very, very similar to mitosis. So this is also known as reductive division because the cell actually gets reduced at this point from diploid to haploid. So this is where we're going to go from 2 sets of everything to just 1 set. And the end result of this, we had 2 daughter cells in meiosis 1. We'll have 4 haploid cells produced here. So we still will call these daughters, but they're haploid. And again, we have that PMAT sequence, but they'll have a 2 after each one to denote meiosis. Okay, so again, just to recap at this point, we have replication during interphase. You end up with the homologous chromosomes with a double set. They each have sister chromatids. And down here in meiosis 1, we're going to separate those homologs from each other. And then in meiosis 2, the individual sister chromatids are separated. So this eventually leads to us having those 4 haploid cells. Again, meiosis 1, you end up with 2. They're still diploid, and then you go to the haploid. Okay, so individual sequences here, meiosis 1, prophase 1, a lot of these hallmarks are the same. I'm going to try to point out some differences here. So right away in prophase 1, a big difference is the fact that we had those homologous chromosome pairs. So again, these are basically double sets of chromosomes that are similar. So they will pair up, they have similar genes and similar size. And again, one is from mom and one is from dad. But a big thing that's going to happen during meiosis 1, prophase 1 is this crossing over, where they will line up and they're actually going to swap parts of their genes. And then the other thing, of course, is our spindle. We still have a spindle, all the machinery is the same, is going to become attached to our homologous chromosomes. So looking at crossing over in a little more detail, what this means, again, our chromosomes were already replicated during interphase, and they already have their sister chromatids. They'll actually line up together and form what's called a tetrad because there's 4 chromatids. As they pair up, they will actually, a portion a lot of times will actually break off of one and the other at the same place. And as you can imagine, this is facilitated by enzymes. And these are going to happen and break at what we call alleles, which are just different versions of the same gene. And when these swap, you can see here that we still have the same overall number of the chromatids, but now they look different. So this leads to new gene combinations and gives a lot of variety in genetics. Okay, so moving right along, prophase 1 was crossing over. Now metaphase 1. Metaphase, you know, they line up at the middle. But I do want to point out here, unlike in mitosis where we had just one continuous line, there you're going to have the homologous chromosomes paired up across from each other. And the other thing you can notice here, because these have already gone through crossing over, these don't look all the same because they've already crossed over and there's a mix-up here. But as I've said here, they don't stay in a line. They're across from each other because these are completely separated chromosomes. Okay, then anaphase 1, you can probably guess, they pull apart. But here, notice it's the homologous chromosomes, so it's 2 full X's being separated from each other. They are going to go to opposite ends. And then telophase 1 in cytokinesis, same thing you would expect. The homologous chromosomes, and again, these look like full X's, they'll go to opposite ends, get new nuclear membranes, cytoplasm divides with cytokinesis, And I do want to point out here that these are diploid still. It's 2 sets still, because if you look, you have 2 sets of each chromosomes. And this is going to be a mix now because crossing over has already happened, and this is now the end of meiosis 1. Okay, so meiosis 2, the events this is going to be like deja vu, because the events of meiosis 2 are pretty much identical to mitosis, but the only real difference here is we do not replicate DNA again prior. Those 2 diploid daughter cells that we just saw at the end of meiosis 1 will each undergo a round of cell division themselves, and this gives us our 4 haploid cells. So again, haploid meaning half, they'll have half the number of chromosomes. And another big difference with mitosis, these are not going to be identical to our original cell. And we'll see that in just a second. So again, at the end of meiosis 1, we're going to have 2 cells that they're diploid still because they have 2 full sets of the chromosomes. In meiosis 2 prophase 2, same kind of things. These are going to become attached to the spindle. At metaphase 2, they line up at the metaphase plate, and then you noticed here, this looks just like mitosis. They're all in a straight line. How you could identify this isn't mitosis, though, you can see it pretty well in that one. Again, they've crossed over. They don't look identical anymore. They're all mixed up. Anaphase 1, the individual sister chromatids separate, and then finally telophase 2 and cytokinesis. And again, this is meiosis 2, that's why we have the 2s after all of them. Then those individual chromosomes are separated, but again, you can tell that this is meiosis because they're all mixed up. Notice this is happening twice, which is how we end up with 4 cells total. Okay, it's almost time for the summary. So with that, we do have just a little video to summarize this process, and that's the end of the lesson.
The cell prepares for meiosis just as it does for mitosis. But as the DNA condenses, each chromosome finds its homologous counterpart. These matching pairs of chromosomes hold equivalent genetic information, one set from mother and one from father. Next comes the first shuffle of nature's genetic deck. The 2 homologs trade genes in a process called crossing over. This forms the hybrid chromosomes. The spindles then place maternal and paternal chromosomes randomly on each side of midline. It is another shuffle that can lead to a host of different genetic outcomes. Finally, spindle fibers pull the homologs apart. This leaves the sister chromatids intact. With twice the amount of genetic information the cells will need, nature must cut the genetic deck in 2. The second division produces 4 unique sets of chromosomes. Because they carry half the genetic information, we call these haploid cells. Many of them will mature into sperm and eggs, the reproductive cells called gametes.
Okay, at this time we are at the end of the lesson, so you do need to write your complete summary and answer the practice questions.